We have a way of showing you that our students have acquired some skills, and we will need to showcase some of those skills. On this note, I would invite the Deputy Dean of Postgraduate School, Professor Ogunshola, to anchor the student session of the program.
We want to appreciate your patience this morning since the beginning of the program. And as we are moving towards the end of the program, we want to quickly bring in some of our students. We have selected just to give us like a sample, especially for new students, so that you will see a sample of what is expected of you. A master's degree work and a PhD work. Those have been selected from those that are recommended from the faculties. So we quickly take that just now. Let me quickly invite the MTech students' presentation. Elegbede Joseph A. Please come and quickly give us your presentation. Thank you for your patience.
Good afternoon. I stand on the existing protocol. My name is Elegbede Joseph Adetunji, and I'm here to present an MTech thesis, valorization of concob by fungal isolate for production of xylanaceae and biotechnology applications as novel biocatalyst. Xylan are polymers that are found in cell walls of plants and algal species, constitute about 20 to 40% of most agricultural waste. They are found in between the lignin and cellulose in the cell wall. The polymerization of xylan, breaking down of xylan, will result in conversion of the xylan fibers to short sugar residues such as xylose and xylooligosaccharide, and this depolymerization can be achieved by the application of enzymes such as xylanaceae. And there are various applications of the enzyme xylanaceae, and these are listed on this slide. The problem is just that concob, which is one of the major agro waste in this country, constitutes a lot of environmental problem because they are just deposited on the farmland after the corn has been removed, and sometimes they are born, which constitute to global warming. And the justification of the study is that the bioconversion of this concob was achieved through the biotechnology production of xylanaceae, which was applied to improve the dough rising and clarification of fruit juice. And the xylanaceae was also applied for biosynthesis of nanoparticles. The aim of the study was to screen fungal strains that are able to produce xylanase on concob substrate under submerged and solid state fermentation, and to examine their biological applications. And these are the objectives of the study. Under this method used, about 8 fungal strains were used, while 4 bacterial strains were also used. The concob was the moisture content of the concob was also examined, and the inoculum was prepared, which was used the inoculum for the organisms was prepared, and this is a flowchart of the qualitative enzyme assay. That is the ability of those fungal strains to produce the enzyme which was first observed. Then the organisms were applied in a corn cob substrate under submerged and solid state fermentation for the production of the enzyme. And the amount of the enzyme produced by each of the fungal strains were examined through the quantitative enzyme assay. The effect of temperature, pH on the enzyme was also examined. Also, the production of the enzyme was optimized using the Taguchi optimization technique, and the aflatoxigenic conditions of the enzymes were also checked, because for the fungi to be applied in food production, we must ensure that they don't produce aflatoxins. And these are the biotechnology applications of the xylanase. The first was in application in bread making, that's in dough rising, while the second is in fruit juice clarification. And like I said, the xylanase was applied for synthesis of silver gold and silver gold alloy nanoparticles. They were characterized, and the nanoparticles were applied for as an antimicrobial. They were also applied in dye degradation process. Also, the antioxidant properties of the nanoparticles were examined, and they were also applied as thrombolytic and blood anticoagulant agents. These are the results of the research. This is showing the macroscopic and the microscopic feature of the fungal isolate that were isolated from sawdust. And this slide is showing the ability of each of the fungal strains to produce xylanase on corn cob media. Also, this slide is showing us the levels of production of xylanase by each of the fungal isolates, and we saw that it was about 10.38 to 50.55 units per mil, which is quite higher than those that have been reported in some previous studies. Also, the xylanase was applied. The fungi was applied for production of xylanase in solid state fermentation, and 50% of the test isolate produced better activities more than the submerged fermentation. For the effect of pH, of temperature rather, on the xylanase, the temperature range of the enzyme produced was between 50 and 70 degrees Celsius, which make it a good enzyme for industrial applications. Also, the effect of pH, the range of pH range from 5 to 7, which make the enzyme produce very good also for industrial application. These are the results of the Taguchi optimization. Initially, when it was optimized, the result improved. We have 62.43 units per mil and 59.16 units per mil. It is quite higher than the conventional method. And finally, when the optimization was validated, we had about 88.39, which is about 70-something percent, about 75% rather, 75% increment more than the conventional method that was used in the production. The result of the aflatoxigenic test showed that none of the fungal isolates that were used produce toxins. That means they are good enough for application in food industry. Now, this is the result for the the xylanase in dough rising. We realized that application of the xylanase improved the quality of the dough, improved the rising of the dough, even better than the conventional yeast used. And it led to about 2.2-fold increment. Also, in the fruit juice clarification, the xylanase improved clarification of fruit juice to the extent of 74.22%, showing that they are very good in this various application. This results shows the potentials of the xylanase in synthesizing silver gold and silver gold alloy nanoparticles, and they conform to some previously reported reports. And this is showing the UV absorbance spectrum of the nanoparticles prepared, also the FTIR of the nanoparticles prepared. The characterization using the transmission electron microscopy showed different sizes of the nanoparticles prepared. For silver nanoparticles, the size range between 15 to 77 nanometer. For gold, it was 4.88 to 123 nanometer, while for alloy, it was 46.74 to 444.2 nanometer. The nanoparticles synthesized produced tremendous antimicrobial activities against clinical isolates. The silver nanoparticles produced antimicrobial activities to the extent of 88%, while gold nanoparticles 44.3%, and the combination of gold and silver alloy nanoparticles to the extent of 90.7%. Also, the range of the antifungal activities of the nanoparticles was very high. It was up to 87.9% by the gold and silver alloy nanoparticles. In the degradation of dye, the nanoparticles degrade malachite green dye to the extent of 92.3%, which was obtained by the gold nanoparticles. Other ones by the silver was 78.97, and the alloy was 91.39. These are tremendous activities displayed by the nanoparticles. Also, in the dye degradation of methylene blue, the activities was the highest activities obtained was 64.1 by the gold nanoparticle. Also, the antioxidant activity of the nanoparticles were also tremendous. It was quite better than that of the standard antibiotics, the beta carotene and the quercetin. We have IC50 of 37.9%, which is quite lesser than that of beta carotene at 710 microgram per mil, showing that the nanoparticles can present quality antioxidant activities. Also, for the anticoagulant activities, the nanoparticles was employed as anticoagulant, and it showed tremendous activities. It was well comparable to EDTA, which is conventional anticoagulant used in blood analysis. And also the nanoparticles was able to lyse already formed blood clots. This can be a good application in treatment of thromboembolism. A lot of people have this kind of problem whereby the blood will clot in the blood vessels, and it will lead to a lot of health challenges. So the nanoparticles effectively lyse blood clots that has been preformed. To the conclusion, we established the biotechnology management of Concorp to synthesize xylanase in submerged and solid state fermentation, and established the industrial relevance of xylanase in dough rising, that is in bread making, and in orange juice clarification, that is in fruit juice production. And we have novel application for fabrication of silver, gold, and silver alloy nanoparticles with excellent activities. As a matter of fact, this is the first time this enzyme will be used to synthesize these nanoparticles, even till date. And we have a couple of recommendations, and the contribution to knowledge, demonstration of production of xylanase in submerged and solid state fermentations by local fungal isolate. Then we established the best condition for maximum synthesis of xylanase by this fungal isolate through optimization. Also, the potential of the xylanase in bakery process and juice production was established, and the first report of fabrication of nanoparticles by xylanase with wide range of biomedical applications. This research has been published in 5 different international journals with impact factors ranging from 1.8 to 2.85, 5 articles. And also, these are the front cover of those of those publications, the 5 of them. And it has been presented in about 5 conferences across 4 universities in Nigeria, where it won the 3rd best oral presentation at the International Conference of Scientific Research at the University of Ibadan. Thank you for listening.
Can we have a round of applause again?
There is what a beautiful presentation, but something is missing in this presentation. What is it? Did you do this work alone? You did not even acknowledge your supervisor, Professor Latif.
I actually want to appreciate my emeritus professor. That's what I call him. In the person of Professor A. Latif. Please, can you give me a round of applause? Give him a round of applause. Is a mentor par excellence. I'm telling you. I can't. Most of this work, it was designed by him. The collaborations abroad and everything was actually facilitated by him. So please help me appreciate him one more time. Thank you so much, sir.
Thank you. You are welcome. I appreciate my team. I was about to introduce the supervisor of the work when he came up. Yeah. As we are rounding up, going to listen to the vote of thanks or the closing remark, let me quickly invite the PhD sample presentation. And that is going to be by Oladipo Kolawole Elijah.
Good afternoon, everyone. I'm standing protocols. I'll be talking on characterization and comparison of pathogenicity pattern of influenza A virus isolated from pigs. Influenza virus belongs to the family of Octomyxoviridae and is an RNA virus, and is a virus of public health importance because it's a zoonotic disease of health concern. We have three types of influenza viruses. We have A, B, C, and by year 2019 we have influenza D, and swine plays an important role in the variant of influenza virus. Many influenza virus persist in pigs, and there could be emergence of a new pandemic influenza viruses. And influenza virus A has one characteristic: it undergo what we call antigenic shift and drift, which leads to reassortment, giving rise to new strain. Now, studies in the developed world noted the importance of pigs in influenza circulation. So therefore, there is a need for us to investigate the of pigs in our society. What is the justification for this study? I've earlier said that pigs plays an important role in epidemiology, and present there is little or no information about the biology identity and the pathogenic study of currently circulating strain of influenza virus in communities. And because of our activities, we have more piggery farms in the country now. People are relating more with pigs. So the aim of this study is to characterize and investigate the differences in the pathogenicity pattern of influenza viruses isolated from pigs. We have the selected objectives for this work. Materials and method. We have our study site. Samples were collected from Bodija. We have a farm. We have two farms from Ogbomoso here. The sample size was calculated using a sample size formula, and virus isolation was attempted from nasal swabs using this procedure. This is just an egg cardinal used in the lab for the procedure. Then samples that were positive from tissue culture of egg were subjected to molecular identification using matrix gene, for example. And from there, we further analysis to carry out genetic analysis on the sequences that was done. We still have some models that were checked in terms of looking at the 3D structures of the protein of the virus. The virus that's been identified were subjected to what we call intravenous pathogenicity tests for influenza using chicken birds. Then the pathogenicity index 6 was calculated based on the standard that has been established by WHO and others. We have TCID50 was also conducted in order to know the amount of effective dose to infect the rats. From there, we have animals were inoculated with TCID50 and samples were collected at specific days for histopathological analysis. Now, the result shows us that from the HA, we have 25 suspected influenza virus, and they were further characterized molecular biology using the matrix gene, and 2 came out as influenza virus H1N2 and H5N1 from Ogbomoso and Ibadan, respectively, as the way it has shown on the table. And this is just the blasting result from NCBI to indicate the name of the virus as it was shown. And the phylogenetic analysis of the virus indicates that the H1N2 from Ibadan shared some similarities with that of the one from Belgium, and that of Doc from Ibadan has shared from Xinjiang, which is a community from China. So this is just a phylogenetic tree showing the clade at which our virus belongs to for H1N1 and H5N1. Of these 2 viruses shows us that indeed pigs are mixing versus where we can see a lot of strains of virus circulating. And this is in consonant to some of other researchers that have worked on it. Then we move on to predict the 3D structure of these viruses to see if the structure can account for their pathogenicity, and we try to see that some residues are present in H1N2 where some are absent in H5N1, which could likely account for their pathogenicity. This is just one of the 3D structures of the protein that was predicted. And A has actually said that in all these differences in structure could confer some pathogenicity differences, and that could make them not to be responsive to either drugs given to antiviral drugs. And the oldest virus that was demonstrated to be low pathogenic may not be pathogenic in future. Like last 2 weeks now, we're going to hear that from China, they isolated H10N3 from a man, and which has been only associated with avian. Now it has found its way to human. So influenza virus can cross boundary from a host to another host. And this is just a table showing us the intravenous pathogenicity index. This is just a percentage trend of the clinical signs of the bird, giving us that the peak of the clinical signs happened on day 3. And few illnesses were observed from this study. Under the comparative pathogenicity of these 2 viruses in rats, we discovered that we observed hypothermia was observed in the virus, showing that the rats were actually infected with the virus. This is just a graph showing the trend of the temperature of the rat. And the weight also reduced with date post-infection, like this graph is showing it. And this work made us to know that the virus can infect rats without adaptation. There are some strains of the viruses that needed to be adapted before they can infect the rat. So it shows us that the virus has a tendency of jumping to human population without being adapted. And this is in line with some other observations of some other researchers. And this is just a summary, table showing us a summary of the histopathologic characteristics of influenza based on the lungs of the rats were examined at different days to see what damages has been done to the lungs. And this is just a plate of this histopathology at the day 15 day post-infection, showing the normal and the affected rat. This is for H1N2, and we are for H5N1. One of the things most interesting about the study is that the animals were not showing any clinical signs, but by the time the histopathological studies was done, we discovered that damages has been done to the lungs. So it means if such a strain was able to cross into human population, human might not be showing clinical signs, whereas damages would have been done to the lungs. By the time that it would be known, then more damages would have been done before that time. So from this work, we discovered that the influenza virus carry very degrees of lung histopathology changes without prior adaptation. However, rats infected with H5N1 has its own peak at 11 dpi day post-infection, where that of H1N2 has at 15 day post-infection. In conclusion, the isolation of the mixture really emphasized that pigs is a mixing vessel. It can accommodate different variants of this work, and the predicted 3D structure also prove that those two isolates are genetically different from each other, and the result also shows us that H5N1 is a low pathogenic strain from the intravenous pathogenicity index, and we have that the study demonstrated like that rats are permissive to infection of strain of influenza virus obtained from this study. Contribution to knowledge. Strains of influenza virus that are circulating among the pig that could be of pandemic potential has been established. The intravenous pathogenicity has been established. It has been shown that the virus can cause infection without adaptation, and the protein, the 3D structure, have been shown that they are radically different from each other. And we have known where the peak for these topological changes could occur. And part of this work has been presented in a conference at Uganda. One was presented in US, one was presented, second one again in Uganda, and we have another one like this that was presented in Singapore. This is just one of the publications from the work. Now, acknowledgement. I want to acknowledge the staff, all the staff members of the Department of Pure and Applied Biology for the role they have played during the course of this study. The people at the Department of Biology UCH, I want to thank them. And the staff at the Department of Histopathology UCH, I want to thank them. I want to thank International Resource Region 2 CDC for the grant they have given me to take part in this work. EDCTP, I want to thank them. UK, I want to thank them. Marketer Foundation, I want to thank them. Medical Research Council, I want to thank them. And I'm still using this medium to thank all lecturers that have taught me from my first degree, second degree, and third degree in LAUTECH. Thanks for listening.